Uh, Welcome everyone. We have gathered today to have a very important webinar that is being organized by National Academy of Sciences India Delhi chapter and Deen Dayal Upadhyaya College. And we have great pleasure in having Professor LEHN amongst with us. And I thought I will just for the younger generation tell you a little bit about the academy, which was Professor Meghnad Saha was the founder. Professor Meghnad Saha was an outstanding scientist. In 1920, when he was about 27 years old, he, along with Professor Satyendranath Bose, were the first to translate Einstein and Minkowski's papers on relativity. And then, as most of us know, in 1920, he put forward the famous ionization formula, which has played an extremely important role in astrophysics, and he was nominated for the Nobel Prize 6 times. But he was not only a great scientist, but also a great institution builder. And in 1930, 17 years before we got our independence, Professor Saha, along with his colleagues, founded the Academy of Sciences with the objective to provide a national forum for scientific interactions and a forum for publication of research work. Professor Saha was the founder president of the Academy, which is partially hosting today's program. It has many chapters throughout India, and we are part of the Delhi chapter. Among the many contributions of Professor Saha, he played a major role in making Allahabad University a world-class institution. He wrote a very famous book on heat textbook, which is used even today. And he created this academy of which we are all part of. He established the Positional Astronomy Center at Kolkata and was its first chairman. He created the Jadavpur campus of Indian Association for Cultivation of Sciences, of which he became the director. And he was the founder of the Institute of Nuclear Physics. And after his death, it came to be known as Saha Institute of Nuclear Physics. He also created the Central Glass Ceramic Research Institute in Kolkata. It is indeed amazing that one person, Meghnad Saha, could contribute so much to the development of our nation. And that was when India has just got our independence of our nation, and one cannot imagine what more he would have achieved had he not died at the early age of 63. In 1869, the great visionary Dr. Mahendralal Sarkar wrote, "The prevailing backwardness of the country was due to backwardness of science, and that the solution was in the vigorous pursuit of the sciences by original research." Unfortunately, it is true even today. And on 28 July 1876, he founded the Indian Association for the Cultivation of Science at Calcutta, supported by generous public contributions, in which Raman did his Nobel Prize winning work. With this in mind, a large number of webinars are being organized. We are trying to and Dr. Manoj Saxena and Dr. Geetika Jain Saxena, they are both playing a very important role in organizing a large number of webinars for the development of science education in our country. So I am Ajoy Ghatak. We look after the NASI Delhi chapter, and I have tremendous pleasure today in welcoming Professor Jean-Marie LEHN, I hope I pronounced it correct, who is an emeritus professor at the University of Strasbourg in France, and also an honorary professor at the Collège de France in Paris. As you all know, he shared the 1987 Nobel Prize in Chemistry with Professor Donald Cram and Professor Charles Pedersen for their development and use of molecules with structure-specific interactions of high selectivity. In 2002, Professor LEHN founded the Institute of Supramolecular Science and Engineering in Strasbourg. Professor LEHN has received numerous international honors and awards, including the Nobel Prize that I mentioned, among them 1993 Officer of the French National Order of Merit and 2014 Grand Officer of the French Legion of Honor. We are indeed very grateful to Dr. Saxena for making all the efforts for organizing this lecture, and grateful to Professor LEHN for agreeing to give this lecture. May I now request Professor LEHN to deliver his talk on Step Towards Complex Matter Chemistry. Professor LEHN.
Okay.
You can stop sharing your screen, please.
Okay.
Yeah, Rosalind, you can share your screen.
Yes. It is. You see it?
Absolutely fine, sir.
Okay. So can I start? Should I start?
Sure, sir.
Very good. Okay, thank you for the invitation to give this talk in these very strange times, you know, where you do everything through electronics and not being there, which is a pity. I certainly would have been happy to come back to Delhi. I've been many times in India, but in the recent years, in the last 15 years, mostly in the more southern part in Mumbai and other cities in the south. So my talk will be a sort of a general public talk mixed in with scientific considerations on chemistry and science in general, and also showing you some of the developments which we have been looking at, which we have been studying along the years. I took a very general title, which is Steps Towards Complex Matter, Chemistry, meaning that I would like to show you that chemistry is really in a very important area in which contributes to the way in which complex matter has evolved. We have to start very early in the history of the universe if we want really to see all the way what has happened. After the Big Bang, the universe was extremely hot, and at that time there was no chemistry, much too hot. But it cooled down quite quickly. Particles formed, and particles then began to associate and to lead to the formation of atoms. So this first period in the universe is the period of physics. No chemistry at that time. When the universe was cold enough for atoms to form, for atoms to connect to one another to form larger entities called molecules, chemistry started around here in our universe. The age of chemistry is starting. Then, in some regions of this universe, a property appeared resulting from the progressive complexification of matter, making more complex molecules, making layers, compartments, and so on. Out of that, a property appeared, which is life on our planet Earth around this time shown here. And this is the age of biology starting. But evolution continued, and a new property appeared, extremely important one, this property which makes us able to talk to each other and to talk about science. This is thought, represented here by a famous sculpture by a French sculptor, Auguste Rodin. Rodin in English, you would say, Rodin in French. The thinker sitting here on his stone and thinking very hard, as you can see. And this is the evolution towards complex matter from physics to chemistry to biology and to the highest form on our planet, which is thinking, the way we do it. Now, let me first stress one thing. This is the end of my slide here on the right, but that's not the end of the evolution of our universe. The universe will continue to evolve. What will happen on our planet, we don't know. There's very probably, almost certainly, life on other planets, in other places in this enormous universe. But evolution will not stop. And so if we find on our planet this very small point in space and in time, people who think that we should stop all this evolution, it's no good to continue, this is not possible. The universe doesn't bother about us. We are just dust in the universe, but thinking dust, and that is what is important. So we now have to ask a very general question. But what is this universe made of? This slide is not it's a dark slide, but it's intentional. What I want to show is that the present knowledge is that there is 68% dark energy, 27% dark matter. It means 95% darkness we didn't really know about in our universe. But that is not what we, as human beings, as thinking organisms, are today interested in as we talk right now. There's 5% visible matter left. And that is, as I like to say, the matter that matters. That's our matter. That's what we are part of. And that is us who are able now to try to investigate. So, in order to look further into this matter more and more complex, there's an important question we have to address. The evolution of matter has gone from divided here to condensed to organized to
Professor Len?
as a function of time towards more and more complex matter on our planet Earth. And as I said already, in other places, probably too. So, the big question then which comes up deals with how does matter become complex? This is for me the most basic question to ask, because that's the question which also addresses why we exist as complex organisms, thinking organisms. So how did we go? How did the universe, coming from elementary particles, generate a thinking organism? And maybe there are even forms of higher complexity of matter. To answer this question, mankind has invented what we call science. And we may consider three pillars of science. First of all, physics. Physics deals with the basic laws of the universe. Those laws which direct what our universe is evolving, how it is evolving, into what it is evolving, and these are the basic laws on which everything else depends. Biology studies the rules of life, how it works, how do living organisms function. So what is chemistry doing? Chemistry is building the bridge between the two. I mean by that how do general laws ruling our universe end up generating something like a living organism on our planet Earth, a thinking organism on our planet? How is this happening? How did it happen? How is it continuing? The answer to this question lies in one word, very simple word, which we can understand, but we have to give it a meaning: self-organization. That is how it happened. It happened because the structure of our universe is such that it will lead to organization somewhere in the universe. How? That's the question. But it's the driving force which does it. It happened by itself on the basis of the laws which rule our universe. One may even say that this is a cosmic imperative in our universe. We cannot escape it. Organization, self-organization, the generation of life, the generation of thought is a built-in property of the laws of our universe. This self-organization we talk about has to be considered in two aspects because we don't have yet a general law for everything. First of all, it's the cosmic self-organization, the large, grand-scale organization of the universe through gravitational forces, which leads to the cosmic structure. And as cosmologists tell us, it's a structuration of our universe by gravitational forces operating on initial inhomogeneities in density and rates of expansion at very early times after the Big Bang. This is, of course, fundamental, but it's not what interests us today. What we are interested in is a divisible matter. What is doing complex matter? How does it form? How does it evolve? This complex matter, molecular matter, results from the action of electromagnetic forces on the bricks which form this visible matter. So this is structuration, a sort of a random structuration by random combinations of the pieces, the bricks, which make up our universe. So this chemical evolution of matter is a very important consideration because chemical evolution, purely chemical evolution, before any life, before any Darwin evolution, this existed before there was life. But it's difficult to say that one has jumped in one step from particles to life. There must be intermediate stages. There's a self-organization of non-living matter, of an evolution of non-living matter, which precedes Darwinian evolution, which comes later, Darwinian evolution, which is a biological evolution of living organisms, but which itself is also a chemical evolution, because Darwinian evolution and the evolution of organisms is based on information contained in the molecules making the organisms. We will come back to that later. So chemistry is therefore considered within the general universe or world of sciences as a science of the structure of matter, how it is built, and the transformation of matter, how matter can be converted from one entity to another one, from one kind of setup, from one combination of atoms to another one. But there's a third property, which we will see a bit later. So along the years, scientists progressively found out that matter was made of pieces, which were called elements. And as time was going, more and more of those were discovered. And they were sort of forming a set of bricks of matter, which were sort of the endpoints. One could not make them smaller, but there was not much order. It was chaotic. Nobody had a sort of a rule for organizing this until the middle of the 19th century, when several people proposed what this was all about. What was this organization of matter? The most successful, and the person who really left his name to that, was Dmitri Mendeleev, who was the first to propose a logical organization of matter. And in 1869, he wrote what I consider as one of the most famous, important, extraordinary papers in science in general, which you see here. This is the original paper by Dmitry Mendeleev in 1869 in the German chemical journal, Zeitschrift für Chemie. And it is, as you can see here, it's written in German. That's the title, is Mendeleev. It's on the relationships between properties and atomic weights of the elements. Mendeleev had noticed that if you take into account the properties of the elements and their atomic weight, you can organize them in a table. And this is the original table you can see here. That's the original table as it was in this first paper by Mendeleev. And here you can see it's organized. For instance, you see here, boron, carbon, nitrogen, oxygen, fluorine. That's the first row in the present table, how it's organized. And then you have also here, you have, let's for instance see, oxygen, sulfur, selenium, tellurium, and so on. Mendeleev went further. That was a big jump also. He saw that there are some places where there was something lacking, and he put a question mark. And he said, these things must exist. And of course, they were found. So please look at this paper as one of the major conquests of science, triumph of science and mankind, organizing the bricks of our visible matter. Now, this is now called the periodic table of the elements, and that's the way it looks now. Now, all of you have seen that, but I like to insist on it because I find one doesn't stress enough how important this table is. It's not just a table for textbooks. It's not just a table to put in lecture rooms. You have to look at it like the bricks of visible matter. And the table is complete. What I mean by that is that the natural elements which make up our universe are all in this table. And there are no other ones. From the planet Earth, a piece of dust in the universe, to the end of the universe, or whatever you might call the end, these elements are there. And there is nothing, not a new one you can insert. Why can you not insert one? Because it is just not possible to do so. The elements here can be counted, and by looking at the number of protons in the nucleus, and of course electrons around them. And there is here one proton, one electron for hydrogen, 2 protons, 2 electrons, helium, lithium, beryllium. You just count 1, 2, 3, 4, 5, 7, 6, 7, 8, 9, 10. And between 6 and 7, there is nothing else. Between 7 and 8, there is nothing else, and so on. So this table is complete. And if anywhere in our universe there are, let's say, living organisms, they are made of exactly the same thing. This is what visible matter is made of. This is absolutely incredible that before our very eyes, we have the table which tells us what everything in our universe is made of, and there are no others. Of course, there's the end of the table, periodic table, which are artificial elements made in accelerators and so on. But I don't want to consider those. I consider those which make up visible matter, then sort of the natural, if you want to say, up to uranium. So please reflect on this, because that is a fantastic step in knowledge, knowing what everything in our universe is made of. And this is the playground of chemistry. Chemists are playing with these elements, sticking them together to make larger entities and rearranging them to an infinite number of combinations. Now, sorry. This research, this is what is composed of, but we need also a rule to convert these assemblies of elements into one another. This was the law pronounced by Lavoisier in the 18th century when he said, "Rien ne se perd, nothing is lost. Rien ne se crée, nothing is created. Tout se transforme, everything transforms." He had recognized that a set of elements combined, organized in a certain way, can be converted in another one, but they are not lost. They are converted into one another. And this is the basis of the chemical reaction equation. Now, this led then also to the possibility of transforming molecules into one another. So after Mendeleev and the elements of the periodic table, people had realized that these had connections and there was a connectivity between these elements. But these connections, these entities formed by connections between elements, were not just a graphical expression of the composition of matter. Chemical objects have a shape. They are objects like architecture objects. And this was recognized in 1874 independently by two chemists, Van 't Hoff in the Netherlands and Lebel in France. They independently published that elements had a shape and that this started what is called stereochemistry, chemistry in space. Most important because indeed molecules have shapes, and this will be very important for what will come later. So, of course, covering the history of chemistry is totally impossible. It would take months or more with many people speaking. But I chose just a few of the very important ones for chemistry. And so I could have cited more, but the ones I cited certainly were among the most important, and as for the reasons I already told you. So from the atom, the basic bricks, to the molecule, molecular chemistry developed. Molecular chemistry, which resides in the strong combination, the strong linkage between atoms to make molecular matter, and mastering the way in which one can build it up. Let's look at two milestones, just to give two examples. In 1828, a German chemist, Friedrich Wöhler, made in the laboratory this small molecule, urea, composed of a carbon and oxygen, 2 nitrogens and 4 hydrogens altogether. In the laboratory, this was made starting from a substance called ammonium cyanate. Now, no need to know what is ammonium cyanate. It is a substance which can, in the laboratory, and has been transformed by Wöhler into urea. This was what is now called a synthesis, a transformation of matter, ammonium cyanate into urea. Furthermore, at that time, it was very important because it also indicated that one did not need this magic force called vital force, which at that time people were thinking to be required if you wanted to make in the laboratory anything contained in a living organism. Vital force was supposed to be necessary, this magic force. Wöhler, by converting ammonium cyanate into urea, which is contained in urine in living organisms, showed that there is no difference between the structures, the molecules of non-living matter and those of living matter. Just a question of type of molecules. So let's jump ahead now and consider this much, much more complicated molecule, vitamin B12. This very complicated molecule, which we have in our body, has been synthesized, that means built up step by step using carbon, oxygen, nitrogen, hydrogen, a phosphorus, a cobalt, has been built up progressively by the collaboration of two groups, one at Harvard University in Cambridge, Massachusetts, directed by Robert Burns Woodward, the other one by Albert Eschenmoser at the Polytechnical University in Zurich, in Switzerland. These were helped, that's been about 120 man and woman years were here at work. And this collaboration then produced in the 70s of last century, this molecule built up from its elements. It's a very complex one. It was representing at that time the ultimate in complexity of constructing molecules from their elements, from the atoms. So molecular chemistry had reached a very adult, very powerful state, and it has continued. Molecular chemistry is, of course, not finished. Since the 70s of last century, new molecules have been made, new procedures have been invented, new materials have been produced, new bioactive molecules, and all that. So it has continued and it will continue. However, we can say, all right, here is how molecular chemistry develops and is continuing to develop. What else? Should we not look for something else? Also, in addition to molecular chemistry. Let me introduce that in the following way. What you see here is a cancer cell, this blue sphere, and two killer cells. The killer cells in our organism, as we are talking, as you are sitting there, they are running around the organism, and their mission is to find out the cells which have gone bad, which have transformed into a cancer cell. And when they find it, they have to kill it, to destroy it. How do the killer cells know that the other one is a cancer cell? What tells them? If they make a mistake, there's a big problem. Either they destroy a healthy cell, or they do not destroy a cancer cell. Another example, here we have a white blood cell with blue dots. The blue dots are the HIV virus. When the HIV virus hits the cell, then it can infect. What tells the virus that it has reached its goal and now it can infect? These two examples, and I could of course cite many, many others. I could cite vaccines, which we are looking for, of course, in present situation. I could cite antibodies, lots of other things I could cite. But let's just look at this. These objects are defined by a membrane, which defines their shape and their volume and so on. This membrane is made of molecules, and in this membrane are sticking molecules, proteins, which touch when they encounter the other body. In other words, the contact between the killer cells and the cancer cell tells, in this contact, the contact between the molecules in the membranes tells the killer cells that the other one is a cancer cell or is not a cancer cell. So something happens between these two large bodies made of many, many molecules. What is going on? What is going on is something beyond molecular chemistry. There must be a chemistry dealing with populations of molecules getting together and interacting. And this is done trying to build up a chemistry which we call supramolecular chemistry, as a chemistry which lies beyond molecules, which deals with population of molecules which deals with what molecules do when they get together, when they interact. And this field has developed now in the last 50 years now. The three main functions which have been studied is, first of all, molecular recognition. This is the basic function. How do molecules recognize one another? That also determines how a killer cell can find out that the other one is a cancer cell. Two other properties: how do molecules react with one another, or how can molecules carry another entity, through a barrier like a membrane of a cell. So let's have a look at molecular recognition first. How can we represent molecular recognition? Of course, first it requires interaction for binding. The entities which get together, they bind together and they have to have an interaction, otherwise they ignore each other. That's, of course, basic and absolutely necessary. But there is another property which is fundamental in molecular recognition, which is information. Without information, you cannot recognize. So the selective interaction between molecular entities rests on the information present in these entities. And this is a sort of a complementarity in geometry of the objects and in interactions. This type of idea has already been sensed, although not in the interaction state, but in the mechanical geometrical state, in the 19th century again, by a chemist, a German chemist, Emil Fischer, who in 1894 published a very famous paper, a basic paper for everything that happens in living systems, when he said that for an enzyme acting on its substrate, they have to fit together like Schloss und Schlüssel, like a lock and a key. That's a very mechanical view, but it is basically fitting. So this was published in 1894, and you can see he got his doctorate in 1874 at our university. We are, of course, very proud of that. So molecular recognition is fitting together molecular objects. The most important example of molecular recognition rests in the genome. The storage of information in the genome of living organism, the genetic information, which makes the living organisms and gives them their personality, if I may say. And this program is written with four letters, which are shown here. It's a long string on which these four letters are sticking. The four letters are rather simple chemical groups, and chemists have given them names: adenine, guanine, thymine, cytosine. And these chemical groups, they look very much the same when for somebody who's not a chemist. And for a chemist, they are very similar, and also they are not very complicated. Now, you may know that two such strands can wrap around each other and give the famous double helix, which is the signature of genetic program, which contains a genetic program of living organisms. So these four letters make up the information which make a tomato or an elephant or a human being or a rat. Now, this is a storage of information in the DNA, in the double helix, in the strand. How is this information processed? It is processed in a very simple way also, by contact between, by pairing up, by contact between these letters. Adenine undergoes contact with thymine or uracil, slight change, by 2 points of interaction represented by these dots. And guanine can interact with cytosine by 3 points of interaction, represented here. So the molecular recognition occurs by these complementary patterns where two molecules can pair up by two interactions and the two other residues, these informational residues, can pair up by three interactions as shown here, as you see here. So after all, we notice that the genome of living organisms is written just with 4 letters and it's processed just by 2 types of interactions. It's a binary system. And why is it like this? Why is it not more complex? One can discuss that. It's not, I have no time now, but maybe the first thing one can say that is sufficient. 4 letters allows for the pairing 2 by 2, and that's enough to store information and to process it. So one could claim, one must claim, and one can claim that chemistry is also an information science. The storage of information is in the molecular level, and the processing of information is at a supramolecular level by interaction between the molecules. So one may consider chemistry as the science of informed matter. It is the science of the structure of matter, of the transformation of matter, and of the information contained in matter. So just very quickly, how we came to that. My initial motivation had to do with neurochemistry, chemistry of the neural system. You may ask, okay, what's that about to do? What has it to do with what is going on here? What it has to do is that I was interested in neurochemistry and I was wondering how a chemist could sort of contribute to the functioning of the nervous system. And one property was, I thought, accessible to chemistry, which is the propagation of the nerve influx, which carries the information along the nerves and the axon of the nerve. And this is the action potential, which carries the signal along the nerve, along the axon of the nerve. And this signal is carried by 2 simple chemical objects, sodium and potassium ions exchanging across the nerve membrane selectively. So the problem was, if one wanted to simplify it in chemical terms at that stage, 50 years ago, the problem was, how can you selectively bind these 2 entities, because you don't want them to they cannot short circuit, they have to be recognized. How can one bind them and transport them through a membrane? And this is absolutely necessary property to be able to carry selectively sodium and potassium along the nerve and to carry and to transport them across the membrane of the axon. These 2 entities, sodium and potassium ions, are part of a series which is contained in the table, in one of the columns of the periodic table of elements, Mendeleev table: lithium, sodium, potassium, rubidium, and cesium. What is it? Just spheres bearing 1 positive charge and increasing in diameter. So one could say, okay, if we want to recognize those spheres and, for instance, distinguish sodium from potassium, if these are the keys, one has to have locks which are complementary to these keys. And this then means that one has to try to recognize spherical cations and to perform spherical recognition. To this end, a number of substances were developed which could recognize these metal ions, these different spheres. And the ones we developed were these ones, cavities, which sort of say are the lock, having a cavity here inside, and which can be adjusted, changing its size by chemical synthesis to make it from small to larger to even larger, and adjust it so as to have a size corresponding to the size of lithium or to the larger one, sodium, or to the larger one, potassium. And this is indeed the case. We call these cryptands. They have contained a crypt, a cavity, and the resulting object, when the substrate penetrates into the cavity, is then, we call it a cryptate. This was the start of these studies, which led to enormous activity in the area of supramolecular chemistry. We'll come back to that, and in how to understand molecular recognition. Here, using the picture proposed by Emil Fischer, here is a lock, a molecular receptor, and here are 3 keys. And if, of course, these are put in presence of the lock, it's the red key which fits. So we have then a supramolecular entity formed by the entry, the binding of the red key into the lock, into the cavity of the receptor. There were many studies performed in numerous laboratories around the world, and of course, many also in India. So let me just summarize a little bit at this stage. In 1828, we have seen that Friedrich Wöhler introduced, so to say, as a first milestone, the notion of molecular chemistry. Of course, molecules were known, but he made molecules, he converted molecules into one another, and this was the start of molecular chemistry. So using molecular chemistry, one can then synthesize what we may call receptor molecules, which are able to use, which are built on covalent bonds between atoms, and which then can lead to associations. And so these associations are supramolecular ones, which come from the binding of a molecular entity to another one, not necessarily into, but to another one, leading to a supramolecular entity, which can have recognition features. How can they recognize one another? Transformation, how can they react with one another? Translocation or transport, so as to give functional supramolecular systems. So in 1828, one can consider that the definition of molecular chemistry, so to say, and in 1978, I proposed the name of supramolecular chemistry for this area, which of course had started 10, 15 years earlier, but it is then defined. So many people were studying this, and as I indicated, around the world, many laboratories were engaged into studying these properties. Let's have a look now at some, I have absolutely no time to go into further details about this molecular chemistry, but let's have a look at some discoveries, some applications. As you know, basic research is acquiring knowledge. Applied research is about applying this acquired knowledge. So the first important application is that understanding molecular recognition helps in drug discovery, because you realize that the synthetic molecule, a drug, is a key for a biological lock. The biological molecule is a target. So it is very important to understand molecular recognition for the developing drugs. We had also made along the years another type of cryptate, which has a cavity which can absorb UV light in its skin, and in the center is a europium ion which can emit red light through the energy transferred by the blue light, which absorbed the UV light absorbed by the skin around it. This molecule is a sort of a red luminescent label which can be attached to immunoproteins, and has led to an immunosystem analysis system for medical diagnostics is now used around the world, at least in hospitals in Europe. So I would just like to stress also another property, this property of transport, which deals with many, of course, with the many regulations of what goes in and out of a cell. But one I would like to insist on is the possibility of picking up a gene, a synthetic, picking up a gene that is a piece of DNA and binding it by a synthetic vector which helps it to penetrate into a cell and then finally goes to a nucleus and finally generates the protein product corresponding to that gene. So this is very important, introducing new genes and letting them being operated on by a cell, very important for gene transfer and for gene therapy and biotechnology. For gene therapy has been applied to correct for genes in living organisms, in some diseases, and also biotechnology. And I would like here to insist on one thing, the fact that these gene transfer processes, this transport of genes into a cell, lead to genetically modified organisms. And I want to insist on the fact that contrary to what now many people are saying, that let's not make genetically modified organisms, this is totally wrong. We need genetically modified organisms. We already make them and we will use them. And in fact, when I see on a label on a product, guaranteed without GMOs, this is crazy. Doesn't matter. GMOs are not toxic. They don't do anything to you. They are perfectly controlled. And we need them to generate new plants which do not exist yet. Genetic modification allows us to make plants which do not yet exist and at the power of genetic modification, at mankind's power, not at natural evolution, but induced evolution. Another aspect is also making supramolecular materials and especially biomaterials. And I wanted here just to show you one thing which is interesting in that respect. Supramolecular kind of polymers. Polymers, you know, are these what one calls plastics, but one can be supramolecular polymers. And they have been developed progressively. We have published the first paper in that area in 1990. And then it developed. And look what happened. 23 years later, well, let me tell you what happened. First of all, over the years, these supramolecular polymers have been shown to have self-healing properties. Here is a transparent film, as you can see between the fingers of one of my Indian postdocs. He made this film in my lab, and so he cut it with scissors. Somebody else cut it for him, as you can see. And then superimposing the two ends of the cut film and pressing with your fingers for a few minutes, you can again take the film and pull on it, and it sticks, which means that the film has undergone self-healing and you recover properties close to those you started with before the film was cut. Then another very interesting property is that these supramolecular polymers have also properties which static polymers, the normal polymers, don't have. And we proposed in 1990 this concept of supramolecular polymers and published the first example of that. But then it took the work of many people, for instance, Bert Meyer in the Netherlands, and then it was developed into biomaterials. And a small company, Cryptate, shown here, developed a material which could be used for cardiovascular implants for children who have a malformation of the heart and require cardiac reconstruction. This took 23 years to develop, very long, but it was extremely satisfying, of course, to see that, because here is the first little girl who was operated on by Professor Leo Bukiria, so Dominika, a young Russian girl. She was operated at the Bakulev Scientific Center for Cardiovascular Surgery in Moscow on October 23rd, 2013. Now it's 7 years since then. And this was 3 months after the operation. And look, she looks happy. He looks happy too, quite obviously, for good reasons. And you can see that, of course, it took a long time, but it ended up in great progress. It is, of course, understandable that it takes a long time, because when you want to put something into human body, lots of controls have to be done. So many children now has been operated on, not just 10. The same company also developed then pulmonary heart valves, which were implanted into children, and in Budapest, Krakow, and Kuala Lumpur. So this is continuing. And they consider it as a breakthrough in surgical practice. Now let's go back to the mainstream. Mainstream meaning that when you understand better molecular recognition, what happens beyond pre-organization, what you would like to do is self-organization. Can one use what one learns from molecular recognition processes to induce a given system to organize by itself? Molecular recognition directed organization. Spontaneous but information controlled. This is very important. It is spontaneous. It is a result of how it is built, but it is controlled. And it happens through molecular recognition patterns. An example which is quite evident is the way a virus builds up. A virus is a dead object, the one which right now creates havoc around the world is one of those. It is not living. It's a bunch of molecules which becomes living only by using part of the machinery of a cell which has been infected. I use here the tobacco mosaic virus, which was the first one studied and understood. And this is an entity, a supramolecular entity, made up of 2,130 bricks protein molecules, which interact with one another surface to surface, as shown here on the right down here, and which progressively builds up a sort of a helical tower with a hole in the middle, and in this hole is sticking the genome of the virus. Here the virus, this green spiral is this genome of the virus. And this looks like magic, but it's not magic at all. It is just plain chemistry, physics, interactions. It's organic chemistry, structural chemistry, physical chemistry, and it just is the result of the way these objects are built up. They automatically associate on the basis of the information contained in these bricks, these molecules here, and the way they interact. So this is a programmed chemical system. The program is such that the program is contained in these proteins, and they associate and lead automatically to this final object. And there's nothing magic. Present science explains all of it. So let's just have these program chemical systems. The program is molecular, and the operation is supramolecular, which proceeds processing via the interaction patterns, which are sort of recognition algorithm. Let me just illustrate in the one coming slide what people have done in one given area. Of course, the self-organization of functional entities has been very exciting and has been the goal of many laboratories. Let me look at one specific case, which is the build-up of architectures made up of molecules linked together by metal ions. The molecules are molecules able to bind a metal ion. These are the components, the bricks, and the cement, the connections, are due to metal ions which bind these molecules together. This has been applied widely, and I just show you some we have made, which are, for instance, helical, double helical, and triple helical entities, shown here, double helix, triple helix. The yellow things here you see, these are the metal ions which link these molecular strands together. Here is an example of a grid an entity where 3 horizontal molecules, 3 vertical molecules are linked together by, here in this case, silver ions, which hold them at each intersection and build up this nice grid, which automatically forms. Or here, what we call a nanocylinder, where 3 linear molecules, 4 flat ones, are linking together through 12 these little spherical entities, 12 copper ions in this case. This is a copper type of structure. And you see here there are 3 plus 4, that is 7, plus 12, that's 19 components, which when you mix them together, spontaneously, but in a very controlled fashion, assemble to this object seen here. These are just other examples. I don't want to get into details, showing you that one can make many others. And this has enormously developed and led to a lot of very interesting chemistry and many possible applications and so on. Let me insist on one thing, that this type of programming self-organization is also of interest for nanoscience and nanotechnology. Indeed, the spontaneous generation of supramolecular architectures, of well-defined architectures which have special functional properties, is of interest because it could be, let's say, not an alternative, but more a complement to the present day need for fabrication or manipulation. Presently, we make all our fantastically powerful devices, computers, phones, and so on, by fabrication, by building these devices. One could hope that if one understands better how to program self-organization, that one might be able to go from fabrication to self-fabrication. This would be the ultimate fabrication. Making objects, making bricks, which are so constructed that they can spontaneously aggregate, organize spontaneously, but in a controlled way, so as to generate the final functional object. Now, I would like now, in just a couple of slides, show you how this has then continued. This gets a bit complicated, so I will just give you ideas, show you what happens, and more describe what happens than going into the details. Up to now, I have insisted on the fact that design using information and programmation of the system leads to the controlled generation of functional molecular and supramolecular entities. This is, of course, molecular chemistry, supramolecular chemistry by design. What about another step in further long complex systems? This is using also selection. In other words, trying to develop systems where the interaction between the pieces are able to select the best pieces for building something up. In order to have an object formed by selection, you need, of course, the system to have enough diversity so as to be able to select the correct pieces to build itself up. And also dynamics, meaning by that when the bricks interact, they may collide and give a sort of a bad combination, and one which is not so stable, so that they have to have the possibility to dissociate a little again and reassociate maybe differently. So selection implies diversity and dynamics in their build-up. This led to the idea of what I call a constitutional dynamic chemistry. That is to say, a chemistry of objects whose constitution, whose build-up is not static, but is dynamic. An object which can fall into pieces, which can dissociate into its components and then reassociate, but maybe differently, so as to be able to respond to agents which come from the medium, either physical, like heat or pressure, or chemical, like different kind of chemical objects. Because these systems can adapt to surroundings, to what happens to agents which are applied, this adaptation property leads to the consideration of an adaptive chemistry, which is now our major field of activity. Setting up entities, systems, which have the ability to adapt to changing conditions. And this has then been applied to the search for biologically active substances by procedures which are the result of these adaptation possibilities of the object itself, dynamic nanostructures, and also dynamic materials. Let me show you, just as an illustration, the behavior of such dynamic materials. Of course, this adaptation has been of much interest also for developing new ways of making drugs, for instance, new drug developing procedures and so on. So let me just illustrate this. Here you see one thing, which is that this material can make polymer which contains, I don't want to go into the chemical details too much, but which results from the connection of one entity here, a dialdehyde, with another one, and generates a chain which is formed by these initial objects. But this chain is built up by the condensation of this amino group with this aldehyde and forms this, what is called an imine bond. In a collaboration with a Japanese company, Mitsui Chemicals, researchers from Mitsui were coming to my group in Strasbourg to study the basic chemistry, and then trying to see what later on, what kind of applications can be made. And here is a case where they developed a film of this kind of dynamic polymers, which then has the following property. First of all, this bond here can be cleaved by water, and this part here can be biodegraded. So this is a sort of, you may call that dynamers are dynamic polymers which have two disintegration pathways, one through chemical cleavage and another one through biodegradation. You may call that a sort of green plastics. Another example which I would like to show, and which is quite interesting, and especially because you see it, is can one introduce changing optical properties because the polymers in question are dynamic. I illustrate that the following way, and please follow me, I'm trying to simplify. They have one film made of two components AB, that's a film, a sort of a plastic film, yeah? Then you have another one made of two components again, A prime and B prime, and you superimpose them by one corner. Of course, this superposition here, if the bond between A and B, between A and B prime can break, of course, A can combine with B prime and B can combine with A prime, which means at the place where they are in contact, two new combinations may appear, AB prime and A prime B. If one of these two combinations has a color or fluorescence, you will see it. And indeed, you see here, my Japanese co-workers, they produced this cat head. This is the cat, one film, AB, that's the ears. And then the inside of the ears, the eyes and the mustache are polymer A prime, B prime. And what happens then? When you heat it up and let the reaction occur, you see that color forms only were superimposed and fluorescence only were superimposed. This means simply that one of these two new entities which have been produced at the places where superimposition of the two polymers was present, one of them is colored and fluorescent and is produced at the superposition locations. So that's an example of what dynamic polymers offer for future development and new properties which are not present, of course, in classical static polymers. Let me at this stage summarize what I said, and then I want to finish with some more general slides. First of all, chemistry starts with being molecular. We have to learn how to build complex molecules, designed molecules step by step from the elements, from the atoms which makes them up. The interaction between molecules then leads to supramolecular chemistry, which leads to organized objects on the supramolecular level. Then, because supramolecular chemistry is based on rather labile bonds, they are dynamic and they can be adaptive. And this is progressive steps towards complex matter. Of course, a lot of work is going on in all these areas from bottom to top here. And of course, even more and more complex forms of matter are being produced. So I hope that you have realized that I want to make you sense that chemistry is able to create novel expressions of matter. Not just what is already around us, but making also, in addition, things that do not exist yet. So the book of chemistry must be written in addition to just reading it. Reading means understanding what already exists, all these complex objects. But chemistry can also write new expressions of matter. And the score of chemistry must be composed in addition to just being played on the score which already exists. So chemistry has enormous creative power. It's a creative science, which again can be represented by a sculpture of Auguste Rodin, the same artist who I already mentioned earlier. And here the hand of the artist expresses out of a stone a sculpture here which is not contained in the stone. It is the hand of the artist which fascinates here, which makes this sculpture, which gives it a shape, which shapes this sculpture. Chemistry, the same way, is the art of matter. Chemistry can produce new expressions of matter which didn't exist before they were made. Now, this kind of idea of the power of chemistry, but especially of science in general, was already sensed about more than 500 years ago, since the 500th anniversary of his death was last year. And this is Leonardo da Vinci. All of you know, of course, the famous painting of Leonardo. And he has written, he was a painter, of course, but he was also a scientist, and he was an artist, he was an engineer. So he had these three features: scientist, engineer, artist, who painted the Gioconda. What he wrote was where nature finishes to produce its own species. That is what exists already in nature. Man begins using natural things. We now know that that is the periodic table of elements, the bricks of matter, in harmony with its very nature. These are the laws of physics. That is the harmony. Like you have to build it to do this by by following the laws of how matter forms, how elements of matter interact and so on. And then comes the ending. The ending is extremely strong for an artist. He was a scientist, as I said. He was an engineer, but he was also an artist, and especially is more known for being the artist. And the ending is very strong, especially when you are an artist, to create an infinity of species. Leonardo da Vinci realized that mankind can produce, can create entities which do not yet exist. This was, of course, fantastic foresight for somebody at these times. So, just some illustrations. You may know that there are a lot of mythologies or myths around the world, in India too, of course. And this is one from Greek history, where Greek mythology, where Prometheus, a god, stole the fire of knowledge from the gods. And here is running away, carrying it, and he gives it to mankind. And here he is showing this and holding high this fire of knowledge here. What is this? This means that mankind has the possibility to gain knowledge, but knowledge we have, knowledge we get, we cannot give it back. We have it, we cannot erase it, and we have to live with it. That is something that gives us, knowledge gives us the power, but we have to know how to handle this power. And our path leads us from looking for more knowledge, the quest of knowledge, to the control of our own destiny. We are now in charge of evolution. We can be, and I'm convinced that we will be more and more in charge of our own evolution. In the past, this information transfer between generations happened in this way. You see here, from the ape to the human, to the modern man, in the present, by this touching, of transmitting information, transmitting knowledge. And in the future, what could it be? Maybe this, where we shake hands, not necessarily with a robot, but I mean by this here is that knowledge, information will be contained, will also be used to transform ourselves. And in fact, we already have done that. People who have a new lens in their eyes, people who may have some metal in their hips or in their teeth, and even more so, if you get a graft, a heart grafted, this heart is not yours anymore. It's a heart from somebody else. Any grafting of an organ is a new organ, and it's not yours anymore. Transplantation creates a sort of a human being which contains a piece from another human being. And in the future, we will more and more develop this sort of hybrids between human beings and objects which human beings have introduced into themselves. Let me finish by citing a mathematician. David Hilbert is a very famous German mathematician who is buried in a city called Göttingen, University City in Germany. Here is his tombstone, and he wanted to have written on his tombstone 2 sentences. What are these sentences? There's one on top and one on bottom. The top is, wir müssen wissen. We must know. That's already a very strong statement. We must know. strong statement. That is also what pushes us scientists. But the second part is even stronger. Wir werden wissen. We will know. This is a vote of confidence. We must know, yeah, but then we will. That is indeed conviction and assurance for scientists. We must know. And then the confidence that we will know thanks to science. And here it is, again, Prometheus, showing high the light of science and of knowledge, which will lead us to know better and better what is surrounding us, what we are, what we represent. And to the young people, I say, science shapes the future of humanity. Please participate. Another aspect of great interest is that science is also without borders. Science has no country borders, no flags, no dogma. Science is not a religion. Science is not, it's not imposed. It is across borders. It is valid for everybody on our planet. And the science is valid for other living organisms on other planets. So science has no borders, and this is one of these properties which, especially for young people, has to be really realized what it means. It means that scientists know people everywhere around the world, countries, and there are no borders between scientists. Thank you very much for your attention. I was glad to speak with you, to give this lecture, and to present some aspects, which are very general, I admit, but maybe from time to time, when I try to think also in conceptual terms. Thank you very much.
Thank you very much, sir, for sparing more than 1 hour, 30 minutes with our participants.
It's my pleasure. It's nice. Yeah. I would prefer to be there, of course, but okay.
That's true. That's true. I mean, we also would have been lucky enough that if you would have been here, but I hope that maybe next year, once the pandemic is over, and if we can arrange for your talk.
I hope so too.
Yes.
But
yes.
Let's wait to be there. Huh?
Absolutely. Absolutely.
Okay.
But so nice of you, sir. I request Professor Khatak.
I would, of course, first like to thank Dr. Manoj Saxena and his colleagues for organizing this wonderful lecture, and our sincere thanks on behalf of all the participants to Professor Len for giving this wonderful lecture, which is inspiring, and the beautiful slides that you have shown. It's really very, very nice. And I'm, as Manoj has said, that we are grateful to you for taking the time. And this COVID crisis has brought a large number of people together through this ingenious technology, and we are grateful to that, and for this technology which enables us to organize such brilliant lectures. Thank you very much, sir, for taking your time and giving this lecture so patiently. And I do hope we will meet in India. The Taj Mahal is now open, and you must come, and it will be good for the younger generation to listen to you in person. You have done so much of great work, and we look forward to that. We look forward to that, and I hope this crisis is over very soon. And thank you again. Thank you, Manoj. Thank you, everyone.
Yeah, you noted that, in fact, 50 years ago, the same crisis would have been much worse because it wouldn't have had all these means of communicating.
Yes.
We can talk to each other, we can see each other, we can correspond with many people. And even this crisis has opened us to, maybe it has also helped us in some way to get better organized.
Yeah, it's organizing such brilliant lectures. I mean, we could never think of 1 year back, but now we hear a Nobel laureate like you and such an outstanding scientist. It's really great and across the country, there are participants in this from Korea. It's a one world. There are no borders now. There are no borders now. Thank you. Thank you very much.